Good morning. In this session, we are going to solve more practical exercises related to the photosynthetic gas exchange. In this experiment, two test tubes were tightly closed. Each tube contained a photosynthetic green leaf, sodium bicarbonate solution as a source of carbon dioxide, and cresol red, which is pink in color. Note that cresol red is a pH indicator. It changes its color depending on the pH of the medium. It gives pink color in neutral medium, red color in basic medium, and yellow color in an acidic medium. Moreover, we know from the previous session that the carbon dioxide makes the medium acidic when dissolved in water. Tube 1 is exposed to light. Tube 2 is placed in darkness. After some time, the pH indicator becomes red in the first tube and yellow in the second, as you can see in this figure. How can the obtained results be explained? Please pause the video and think about it. Now, listen to the explanation, and then, correct your answer. At the beginning of the experiment, the pH of the medium was neutral and the pH indicator was pink in color. In tube 1, the plant is placed in light, so, it undergoes two biological processes, which are respiration as well as photosynthesis, but of course, the photosynthesis rate is much higher than that of respiration. As a result, more carbon dioxide will be absorbed by the plant than released, and as a consequence, there will be an overall carbon dioxide absorption. But in the second tube, where the plant is placed in darkness, it only undergoes respiration, because photosynthesis cannot occur in the absence of light. As a result, carbon dioxide is released by the plant, and, as more carbon dioxide gas is produced, the medium pH will change from neutral to acidic. Of course, the answer to this question should include all these information, as follows. In tube 1, where the green leaf is exposed to light, photosynthesis occurs, while in tube 2, photosynthesis cannot occur due to the absence of light, so, only respiration takes place. As a result, carbon dioxide is absorbed in tube 1 to be used for photosynthesis, and, in the absence of carbon dioxide, the medium will become basic, consequently, the cresol red turns red. However, in tube 2, the medium became acidic due to the production of carbon dioxide by respiration, and thus, cresol red turns yellow. Another experiment was done, in order to study whether photosynthesis is affected by light color spectrum. A spirogyra filament, which is a filament of a chlorophylltic green algae, is exposed to a light spectrum, including violet light, green light and red light. Aerobic bacteria, which require oxygen to survive, were grown nearby the spirogyra, and are then observed under the microscope, to detect the bacterial growth and multiplication. The bacteria are found to be differently distributed around the spirogyra, as shown in the figure, represented by this curve. First question, explain why the bacteria approach the algae. Second question, specify whether the bacteria would behave in the same way in darkness. When they ask us to specify, this means, to indicate and justify. Third question, draw out a conclusion based on the distribution of the bacteria. Fourth question, explain the observed results. Pause the video, and solve this exercise. Now, let us correct the answers. For the first question, which is to explain why the bacteria approached the algae. They have told us in the given that the bacteria used is aerobic, which means that this type of bacterial can only grow in the presence of oxygen, as you can see in this figure. The bacterial cells multiplied at the upper level of the tube, where the oxygen level is high. And since the spirogyra is a chlorophylltic algae, this means that it can undergo photosynthesis under specific favorable conditions, and produces oxygen. Therefore, the bacteria approach the algae to take the oxygen produced by the plant during photosynthesis. The second question is to specify whether the bacteria would behave in the same way in darkness. Of course not, because photosynthesis requires light, and in darkness, photosynthesis does not occur, so, oxygen is not produced, and as a consequence, the Spirogyra will no longer be a source of oxygen for the bacteria. In question 3, we have to draw out a conclusion based on the distribution of the bacteria, that is, we have to refer to the observed results, but there is no need to analyze, we directly write the conclusion. In this case, we have to think about the following. This curve in the figure, next to the Spirogyra filament, represents the number of bacteria, and of course a high number of bacteria means that the bacteria multiplied and grew in number. So, we can say that, during photosynthesis, oxygen is produced, and as a result the bacteria will use this oxygen to multiply. And we notice that, when the plant is subjected to either red light or violet light, there was multiplication of bacteria, but, when it is exposed to green light, the bacteria could not multiply. Therefore, the green plant can use violet or red light to undergo photosynthesis, however, it cannot use green light. Concerning the fourth question, we were asked to explain the observed results. So, in the answer, we have to write in details all what we have previously thought about, and which have helped us to reach at the conclusion. In this experiment, in the presence of either red or violet light, the Spirogyra undergoes photosynthesis, resulting in the production of oxygen gas, which is needed by the aerobic bacteria. As a result, a bacterial colony is formed nearby the algae at these regions. However, in the part that is exposed to green light, photosynthesis cannot occur, thus, oxygen is not produced and the bacteria does not approach the algae at this region due to the lack of oxygen.